Welcome to Zeo Skin Health's webinar on the role of DNA and aging skin, presented by esteemed Zeo faculty member Dr. Kristin Tarbet. My name is Deborah Toms, and I am the Vice President of Zeo Skin Health Account Business Development and today's webinar moderator. I'm excited that we have hundreds of participants from all over the world joining the webinar today. In an effort to better serve our physicians and professionals, Zeo will continue to host a series of educational seminars and webinars based on Dr. Zain Obaji's latest clinical findings and his scientific thinking. For more information on upcoming events hosted by Zeo Skin Health, please visit the Zeo events page at zeoskinhealth.com. Now, before we begin, I do have a few important points to review. First and foremost, know that we are coming to you live from Seattle, Washington, where it is very windy and very rainy, and power outages have been known to go out throughout the county. So there is a possibility that we may lose power during this webinar, but not to worry. We will record the webinar and we will get it out to all of you who have signed up. But first, please know that all participants are muted for the entire webinar. Second, participants can type questions into the webinar chat box, and Dr. Tarbet will answer as many as possible at the conclusion of her presentation. Note that you can type questions anytime during the webinar. Lastly, this webinar may not be copied, recorded, reproduced, or distributed, as this material is the intellectual property of Dr. Kristin Tarbet and Zeo Skin Health Incorporated. I'd like to introduce you to today's guest speaker, Dr. Kristin Tarbet. Dr. Tarbet is an oculofacial plastic surgeon in practice in Bellevue, Washington for over 15 years. She is in private practice and owns and directs Amara Medical Aesthetics. She is author of numerous scientific publications and textbook chapters, as well as a national instructor and lecturer. She is an educator serving as a clinical professor at the University of Washington, as an ASO PRS fellowship preceptor, and a Zeo faculty member. Today's webinar topic is the role of DNA on aging skin, and I am pleased to introduce to you our host for today's webinar, Dr. Kristin Tarbet.
Good afternoon. It's my pleasure to be able to speak with you today on the role of DNA in aging skin. Let's start the foundation for our discussion this afternoon. Our skin is the largest organ of the body and also the easiest organ for us as physicians to examine for evidence of disease. It is also the organ that conveys to others our health and our vitality. So what is the function of this, our largest organ? It functions to protect our inner organs and maintain homeostasis, to conserve body heat through insulation and vasoconstriction, as well as to cool the body through sweat evaporation and vasodilation. It functions to relay information to our brain about our external environment. It synthesizes valuable vitamin D, and it carries out antigen processing and immune surveillance. It also prevents invasion of water, bacteria, and other areas through a healthy, strong barrier function. Our skin is an adaptable organ. During our lifetime, the skin undergoes numerous adaptations. This begins at birth, when it is removed from a fully aqueous environment to exist in an environment largely made of air. Throughout our lifetimes, our skin undergoes numerous hormonal changes, trauma, various types of stress such as disease and environmental, and of course, our aging changes. So, what is healthy skin? The definition of healthy appearing skin is characterized here, and I think we'd all agree it denotes smooth, firm, tight skin that's even in color, even in texture and contour, hydrated, tolerant in its function as a barrier function, and free of clinically apparent disease. In order for skin to appear healthy, the cellular functions that provide for healthy skin must be working optimally. Truly healthy skin can be achieved through the philosophy and practice of the Zeo Skin Health Circle. This circle represents a continuum wherein prevention, treatment, and maintenance are addressed as core objectives in skin health, in a comprehensive and continuous regimen. ZEO Skin Health Restoration and Maintenance is made up of these 6 principles. For our discussion topic today of the role of DNA in skin aging, I would like to focus on the 3rd principle of stabilization and the 6th principle of sun protection. So let's start by talking about the effects of stabilization on our skin. 1. Stabilization involves repairing of DNA, and sun-damaged atypical keratinocytes are shed, and they are replaced by healthy cells. 2. A strong barrier function is restored. 3. Soft keratin increases, and epidermis thickens. 5. The melanocytes become more resistant. And lastly, our healing time and outcomes improve. Skin stabilization occurs at 2 levels, both the epidermal and dermal level. This slide is busy, but divides the 2 levels of epidermal and dermal into columns and lists what is occurring at each level to stabilize or to optimize the cellular functions at that level. On the next slide, we see the products that support and stabilize these cellular functions at these levels that are products of our ZEO Skin Healthcare line. The epidermal stabilization, I'd like to focus on the C-Bright and the Daily Power Defense, and then we'll dermal stabilization, which also includes the C-Bright and continues with the growth factor, the Advanced Radical Night Repair, and Retamax. So let's talk about DNA. DNA carries our genetic code, and healthy, accurate DNA replication ensures healthy, accurate cellular function. So what is going on at the cellular function in aging skin? And how does stabilization minimize and prevent these changes? DNA is made up of 2 complementary strands of nitrogen bases matching up correctly to form a double helix. In this accurate pairing, guanine must match up to cytosine and adenine to thymine. It is very important that this structure and pairing remain intact in order for DNA to replicate correctly and form healthy complementary strands as shown here. Damage to this accurate DNA replication and therefore healthy cellular function results in skin damage. This DNA disruption can come from various factors. Damage to double helix within the cellular nucleus due to dimers, oxidative stressors at the nuclear and mitochondrial level, as well as glycation and alkylation, just to name a few. Intrinsic aging over the years is a result of these normal cellular replicative processes breaking down with time or diminishing in their function. Cellular elimination of oxidized proteins lessen with age, and we also see decreased expression of MMP, as well as innate DNA repair mechanisms, which can lead to heightened cellular damage, the byproducts of this damage, and even a heightened mutation risk to the cells. We will discuss 4 of these DNA repair mechanisms and the enzymes that catalyze and facilitate these repairs. And we'll discuss how, by delivering these enzymes, we can help to stabilize the normal, healthy repair mechanisms and thereby stabilize healthy cellular functions. The first enzyme, OGG1, or 8-oxoguanine glycolase, is responsible for removing the oxidative byproducts like 8-hydroxy-2-deoxyguanine that can cause a mismatch base pairing in DNA replication. These oxidative byproducts have been linked to the development of skin cancers, and the OGG1 enzyme repairs these oxidative byproducts, so thereby supplementing its activity and increasing the healthy DNA repairs. Hopefully, this would also subsequently prevent the development of skin cancers. So, to enhance this OGG1 activity and its DNA repair, roxasomes, which are OGG1 encapsulated within liposomes, and are delivered for topical delivery and are used to enhance the OGG1 actions. Roxasomes enhance the skin's natural DNA repair mechanisms and have been shown to therefore shorten the time for nuclear DNA repair from 24 hours down to only 2 hours. Thus, the natural DNA repair process of mismatch base removal is enhanced by these topically delivered OGG1 enzymes via the roxasomes. These assist and increase healthy DNA replication and thus healthy cell function, leading to healthy skin. Another such natural DNA repair enzyme is UV endonuclease. This enzyme recognizes distorted, mismatched DNA strands due to thymine dimers and facilitates repair and removal of the dimer and thus repair of the DNA chain. These endonucleases also downregulate and reduce MMP levels, cytokine levels, and other byproducts that can accumulate in the cells during oxidative stressors. To enhance this endonuclease function in DNA repair, this endonuclease enzyme is encapsulated in liposomes, just like the roxasomes, but this liposome containing endonuclease is labeled ultrasomes. These ultrasomes can then be delivered topically, thus delivering the enzyme, and have been shown to accelerate DNA repair fourfold. Additional natural DNA repair enzymes include photolase, which reduces cellular apoptosis and cellular DNA damage from cyclobutane pyrimidine dimers. There are, of course, other naturally occurring DNA repair precursors which supplement and boost the skin's repair mechanisms. Amino acids such as proline, acetyltyrosine, and ATP assist, and when supplemented will also hasten DNA repair and therefore reduce this DNA damage. This slide depicts the combination of these mechanisms. The roxasomes containing the glycosylase, the ultrasomes containing the UV endonucleases, and the other enzymes that we just discussed, working in conjunction to assist the natural DNA repair process for the skin. When these DNA repair processes are supported, stabilization of cellular function occurs. At the level of the epidermis, that means a strengthened skin barrier, promoting increased skin tolerance to both internal and external stimuli. It means optimizing natural exfoliation of the epidermal cells. It means repairing the damaged DNA and restoring and maintaining adequate hydration to the skin. It also controls melanin production. The two Zeo Skin Health products that I'd like to focus for epidermal stabilization include the Essential Daily Power Defense and the C Bright 10% Vitamin C Serum. These both provide epidermal stabilization by supporting and contributing to the healthy epidermal cellular functions as listed here. Daily Power Defense does so by delivering the enzymes involved in the skin's cellular DNA mechanisms via the ultrasomes and roxasomes as we discussed. Additional ingredients included in the Daily Power Defense, such as antioxidants and the other enzymes we discussed, also help support healthy cellular function at this epidermal level. Daily Power Defense can be used by everyone and is indicated in all skin types. And it provides the DNA support and stabilization of normal, healthy epidermal function as listed in this benefits listing. C Bright, Zeo's vitamin C serum, also assists in normal cellular function with antioxidants in order to arrest inflammation, as well as 10% ascorbic acid and tetrahexyldecyl ascorbate to both break up old pigment and control the development of new pigment at the epidermal level. as well as at the level of the dermis. Vitamin C, as we can see in this picture here, can penetrate through to the dermis through the texahexadecal to help prevent new pigment formation by up to 80%. This also helps to stimulate healthy collagen and glycosaminoglycan production. So vitamin C is able to stabilize the epidermal as well as the dermal level. Going on to dermal stabilization, dermal cellular functions are improved, including production and quality of collagen and elastin, glycosaminoglycan increase, and improvement in the circulation to the skin. Additional Zeoskin products, such as growth serum, radical night repair, and Retamax, provide varying degrees of aggression and focused treatment to also contribute these products for further stabilization of the dermal level cellular function. With dermal stabilization, additional healthy cellular function is supported and provides further anti-aging benefits, affecting fibroblast production of collagen and elastin, improving and maintaining the skin's texture and tone, and improving circulation, which also serves to enhance epidermal function through this improved oxygen and nutrient perfusion going to the basal cell layer. So we've talked about DNA's role in intrinsic aging, as well as discussing how the process of stabilization with the Zeo Skincare line can improve these healthy processes at the epidermal and dermal levels. Now let's talk about sun-induced skin damage and the DNA damage that it causes, as well as the Zeo sun protective ingredients to combat it. This spectrum of sunlight depicts the various wavelengths of light. We know ultraviolet light, particularly UVA and UVB, can damage our skin repair and our cellular processes. But visible light, particularly HEV, or high energy visible light, in the blue-violet or 400 to 500 nanometer wavelength range, can also induce harmful damage and premature aging. Interestingly, red visible light in the 600 to 700 nanometer wavelengths has been shown to result in elevation of collagen and elastin content, thereby helping to improve our skin texture. HEV light generates the same amount of ROS, or reactive oxygen species, in the cells. It affects the skin's inflammation cascade and accelerates skin aging. But its longer visible wavelength, as seen to the right in this figure, allows the HEV light to penetrate deeper into the dermis than UVA or UVB light. So where zinc and titanium dioxide can block UV rays, only fractionated melanin can shield the skin from harmful HEV rays. A normal melanin cell is depicted on the left of this slide. Melanin is photoprotective, occurring naturally in our bodies. On the right side of this slide is depicted the process of naturally occurring melanin being released from these melanocytes as a first line of defense against exposure to damaging light waves. Fractionated melanin is designed to be used as an additional defense to protect and to shield the skin from these harmful, more deeply penetrating rays of HEV light. To sum up the comparison of HEV light to UV light, basically the important point is that these HEV light waves pass the deepest. In addition, HEV light, by penetrating deeper into the dermis and circulation, may induce accelerated photoaging and carcinogenesis. Although HEV light does not cause the immediate erythema or edema reactions as seen with UVA or UVB, its radiation is associated with less direct DNA damage, but more associated with oxidative stresses and generations of free oxygen radicals. Fractionated melanin is designed to absorb HEV light and scavenge these free radicals, thereby shielding the skin from these adverse effects of HEV light. Fractionated melanin is not designed to absorb the beneficial rays of red visible light. Therefore, the fractionated melanin, by absorbing the harmful blue-violet light, but allowing the beneficial rays of the red visible light to pass, these beneficial rays can travel to the dermal fibroblasts and boost our production of collagen and elastin. This is a slide courtesy of Dr. Obagi to underscore the importance of sun protection at a young age to protect and improve or to boost our healthy skin cellular function and skin appearance from early on. All of ZO sunscreens include this fractionated melanin, which provides a longer-term protection, more on the order of 6 hours, in conjunction with the shorter-term protection of the physical and chemical ingredients contained. In mention of physical and chemical sunscreens, let's further define and describe sunscreens in general. Physical blocks are inorganic blocks that do not contain carbon in their chemical structures. Their mechanism of action is by reflection and refraction of UV light. And the two most commonly used inorganic compounds within these products are zinc oxide and titanium dioxide. For our ZO Skin Health line, this includes the Eclipse Sunscreen and Primer SPF 30 and the Eclipse C Broad Spectrum Sunscreen SPF 50. Both contain physical blocks, whereas chemical blocks contain carbon in their chemical makeup, and their mechanism of action for protection is via UV light absorption through a photochemical reaction. These type of sunscreens include our ZO Eclipse Smart Tone Broad Spectrum SPF 50 and the Sun Spray SPF 50. Again, all ZO sun protection products contain the fractionated melanin and therefore provide maximal skin protection from both UV light as well as the deeper penetrating HEV light rays. Plus additional ingredients and antioxidants that we'll mention later that help to repair and to protect DNA and other skin cellular functions. Let's talk about ZO's two physical sunscreens. First, the Eclipse Sunscreen and Primer, and next, the Eclipse C Broad Spectrum Sunscreen. They both have the additional ingredients listed at the bottom of the page that help to provide further DNA protection and improve skin function. Notice that they both contain the fractionated melanin, as we mentioned, to protect against the harmful HEV rays. But note that the active ingredient at the top is titanium only. Why do ZO physical sunscreens not contain the zinc oxide as well as we typically see in over-the-counter sunscreens? Well, the efficacy of any reflective sunscreen, such as titanium dioxide or zinc oxide, or a combination of both, is based on particle size properties. ZO's new formula contains a larger, more effectively optimized particle size distribution of titanium dioxide. And compared to our prior formula, which utilized a smaller, less optimized titanium particle size, we no longer need to require the zinc to be present in order to achieve the same efficacy profile. So the larger particle size titanium dioxide in the new formula is protective. Our Eclipse Sunscreen, Primer and Broad Spectrum SPF 30 contains the ingredients as we mentioned. The function of this product then is not only to provide the UVA and UVB protection, but as well the HEV protection. The additional ingredients assist with DNA protection and repair, as well as an exclusive 12-hour time-release antioxidant, as well as anti-irritant. It contains a hydrator to replenish the moisture as well as to help restore our healthy skin barrier and lipid layer. The indication again is for all skin types, so everyone can use this product. The benefits include the sun protection, the DNA repair, and improvement in the lipid and moisture qualities of the skin to enhance the skin barrier and to help smooth it and diminish the appearance of our texture and fine lines. Eclipse C broad spectrum sunscreen has the ingredients as listed, including the additional ingredients of antioxidants, vitamin C, E, as well as a third-line defense against cell-damaging free radicals. It also helps boost naturally occurring cell lipids and an anti-irritant to prevent skin redness from the sun exposure. It also contains a unique complex to provide the hydration and lipid layer to support our natural, strong skin barrier function. The Eclipse C broad spectrum sunscreen, which is recommended by the Skin Cancer Foundation, is found to be effective for post-procedural protection as well as all skin types. It's used on the face and the body, provides multiple defense protection, as we mentioned, to UVA, B, and HEV. This ultra-mild formula is suitable for even the most sensitive skin types. It as well contains the anti-irritants and the powerful antioxidants, as well as the lipid and aqueous restoration hydrators to enhance our skin barrier to prevent dehydration from daily environmental exposure. The clinical skin findings of sun damage include early changes such as redness, roughness, dullness, and dyspigmentation of the skin. Later changes include wrinkles, leathery texture, laxity, lentigines, and malignancies. Over-the-counter sunscreens offer only UV light coverage for a short period of time. The ZO Skin Health Circle philosophy of lifelong healthy skincare targets treatment at the cellular DNA level. And the picture of this healthy skin circle here includes maintenance as well as daily skincare, prevention and therapeutic interventions in order to obtain that healthy lifelong skin. The ZO Skin products that we discussed and the comprehensive philosophy of ZO Skin Health concentrates on the prevention and management of skin cell function at the DNA level. The products we have discussed today help in the stabilization and protection of healthy skin cell functions. To summarize, DNA damage and its effect on aging skin can be combated through stabilization and support of healthy cellular processes through effective DNA repair agents delivered through liposomal encapsulation, and also through UV and HEV light protection by both physical and chemical blocks, which provide the short-term or 2-hour protection in conjunction with fractionated melanin, which provides the longer-term 6-hour protection. These stabilization and protection processes will lead to healthier functions of our body's largest organ, the skin, and therefore a healthier and more vital appearance to our skin for a lifetime. Thank you. I see that we've received some questions that have come through despite this blustery weather, and I'll take a minute now to answer each of these. Question number 1 states, how often should one reapply sunscreen since fractionated melanin lasts up to 6 hours? Well, the answer is that we should be, with our ZO products, applying every 2 to 3 hours due to the limited short-term protection of the UVA, UVB protection requirements. All right, question number 2. Where does ZO's fractionated melanin come from? That's a good question. The zeos fractionated melanin is derived from corn, so we haven't had many problems with its utilization, and obviously is one of the exclusive components of our sunscreen that, as we've learned today from the DNA damage that can occur, protects against that, the more deeply penetrating HEV light, which I think is one of the highlights of our Focus ZO skincare health regimen and protection for DNA damage. Question number 3. When would you apply C-Bright, and when do you apply Daily Power Defense? Well, Dr. Abaji has as I've listened in the lectures and in my discussions with him, has recommended, when you're using the two together, to use the C-Bright in the morning and Daily Power Defense in the evening. And that's what I do and what I tell my patients as well. I received another question, when do you start to see the intrinsic signs of aging? Yeah, I hate to say it, but we start to see it early on. And I remember Dr. Abaji once describing it as, by the age of 30, your cells start to go to sleep. So that's a little depressing, but our intrinsic signs of aging start as early as our 30s and early on in our life. That's why that early protection and that showing the slide for sunscreen as well as stabilization in that young patient is such an important concept in ZO Skin Health for lifelong healthy skin. I have another final question that has come in. Is Daily Power Defense safe for kids? Yes. Daily Power Defense is safe for utilization in anyone ages 10 years up, 10 years old and up. So yes, very definitely safe for children, recommended age of 10 years old and older.
Thank you so much, Dr. Tarbet. Very informative, very scientific results, and I really do appreciate you taking this on. So this now concludes the webinar, and we thank you so much for participating. If you're interested in finding out how to become a member of the ZO Skin Health Circle Club, please contact ZO Skin Health through our website at zoskinhealth.com. And if you'd like to purchase Dr. Abaji's new book, which is called The Art of Skin Health Restoration and Rejuvenation, simply visit zoskinhealth.com. Likewise, for more information on upcoming events with Dr. Zain Abaji and his distinguished faculty, please visit the events page at zoskinhealth.com. And finally, for private training with Dr. Zain Abaji at the Abaji Skin Health Institute in Beverly Hills, please call 866-276-2990. And if you have any questions for Dr. Tarbet, you can call her at 425-455-2131. And her website is listed there for you on the screen. Thank you so much. I'm so glad we did not lose power in blustery Seattle today. And enjoy the rest of your day or evening. Bye-bye.